Yo, yo, yo. As promised, I am away, but you'll still be learning from me. Topic 7 Gene Regulation Overview. We have already learned about how genes get expressed. The DNA gets transcribed into mRNA, and mRNA gets translated into a functional protein. And from there, those functional proteins carry out our traits. So a cell can actually regulate to what extent they can express the gene contained in the DNA, and this is called gene regulation. Some genes are expressed more than others, and some none at all. And so again, this all depends on the signal transmission within and between cells and even the environment. These are the important components of gene regulation. So first off, we have the regulatory genes, which is a sequence of DNA that code for either regulatory proteins or small regulatory RNA. Well, with regulatory proteins, and this would be important for the prokaryotic ones, these are proteins that bind to that DNA to either facilitate or hinder transcription of the DNA by RNA polymerase. Then we have the small regulatory RNA, which would bind to mRNA and activate or inhibit the translation of that mRNA. And then we finally have the regulatory sequences. These are non-coding sequences of DNA that regulatory proteins bind to. And then it also includes the promoter, which would be the part of the DNA that the RNA polymerase binds to in order to start that transcription. I'm a very visual person, so seeing all these terms on the DNA itself really helps. So again, we see the regulatory sequence in yellow, and then the regulatory gene is located, and again, this is considered upstream, okay, of the regulatory sequence and the genes that code for the mRNA. And so that regulatory gene produces two things, like I said before, the regulatory protein that binds directly to the DNA, and it either inhibits or promotes transcription, or it produces this small regulatory RNA, and this small regulatory RNA binds to the mRNA and prevents translation from happening. So again, two parts to this regulation of gene expression. So there are two types of regulatory proteins, one that increase and one that decrease transcription of a gene. So we take a look at the negative control, right here, negative control. So again, the repressor binds to it, and then there is no transcription happening. Okay, so these are considered to be the repressors that bind to DNA and block transcription when they are activated. And then we have the positive control, and the positive controls are considered to be activators. And then again, these bind to DNA and stimulate transcription when they are activated. And so again, it looks like they are the same thing, but produce two different results. With that, we are done with the overview and we're ready to dive into the first one, gene regulation in prokaryotes. Prokaryotes conserve energy and resources by making certain proteins only when they are needed. So this makes them very special. They can adapt to environmental changes and either increase the expression of certain genes to produce a certain product they need, or if the product is available, they can inhibit that expression. So for example, we see here in this picture, so we have these three enzymes here that eventually produces tryptophan. But there's a lot of tryptophan in the environment already. Tryptophan can actually, again, inhibit this gene expression, thus stopping the production of tryptophan itself. So the bacteria wouldn't need to create tryptophan. Tryptophan is there, and so it doesn't waste any energy or resources creating tryptophan because it has a lot of tryptophan in its environment. So this is what makes them so awesome. In prokaryotes, a group of functionally related genes can be controlled together by a single on or off switch. And this on or off switch is called the operator. And again, it is a short stretch of DNA that controls the access of RNA polymerase to the genes that, again gets transcribed and then translated into proteins. So this operator is positioned within the promoter. And what is a promoter? Well, it is again a stretch of DNA that the RNA polymerase attached to in order to start that transcription of those genes. And so the promoter, the operator, and the genes that get expressed are all called an operon. Okay, so again, the promoter is where the RNA polymerase attached to start that transcription. Operator allows that RNA polymerase to transcribe those genes, and then genes produce certain proteins. So again, I'm a very visual person, so let's put all those terms together. So the operon, right? The operon includes three things. First off, the operator, which controls, this is the on or off switch, controls the transcription of these genes here. Okay, and then we have that operator within the promoter. The promoter is where the RNA polymerase binds to, and as it moves along the DNA, it will transcribe in this portion here that I am highlighting, right here, to produce, again, mRNA, and that mRNA gets translated into these functional proteins. And again, these proteins, and for this specific example, produces tryptophan. Okay, so again, this part, the operon, is three parts: operator, on or off switch, then the promoter where the RNA polymerase binds, and then the genes. Again, it's a single mRNA that produces many different proteins, as you guys can see. There you go. So we have the negative control of operons. So let's remind ourselves what that negative control means. It means that the operon is switched off by something called the repressor. And the repressor is just a protein that binds to the operator, and it doesn't allow RNA polymerase to transcribe the gene. So this repressor is on that operator. So again, we see that this RNA polymerase cannot go through to transcribe these genes here. Okay, so again, that is the repressor. So the repressor protein actually gets produced upstream. So again, this is upstream of the operon. So these are called regulatory genes, and these regulatory genes create that mRNA, which creates that repressor. Okay, so in bacteria, there are two types of negative control. And this is a key here, and this is where a lot of students get confused on this concept. So again, there are two types: repressible and inducible. And the best way to remember the difference is to use the English language. And I'll expand upon that as we go through repressible and inducible. So we're going to cover repressible operons first. And so let's just read that definition. A repressible operon is one that is usually on and expressing genes. So let's rewind and use English to remember this better. So the repressible means that we can stop it. So we can stop it. So the default for this operon is on. Okay, so that's one way I remember it. And then when we're describing this repressible, we're actually describing what that repressor is doing. So that repressor is not attached to the operator, it is not active, and again, thus the expression is always on. So when it is activated though, and it is the repressor, when that repressor is activated by a small molecule, and we need to know these names by the way, is called the co-repressor. Okay, so that co-repressor activates the repressor, and then that repressor protein can then bind to the operator. And once bound to the operator, the RNA polymerase cannot transcribe those genes, and thus we have essentially turned off the operon. So again, repressible, we can stop it. Default, it is always on. Nothing's in the way of RNA polymerase, and RNA polymerase is transcribing it. Okay, the primary example of this repressible operon is the trip operon. So the bacteria is always producing tryptophan, which is an essential amino acid. And how does it get created? Well, there are 5 polypeptides that create this tryptophan. And again, that is regulated by 5 genes bundled together in this operon. So remember, repressible means default is on because we can stop it, right? We can turn it off, repressible. So tryptophan can act as a co-repressor. Once there's enough tryptophan in the bacteria's environment, it can act as a co-repressor and activate this repressor protein, and then that repressor protein can then bind to the operator, which will block RNA polymerase right here from transcribing the genes. So again, this process is very efficient because bacteria will be able to save their energy, because if they're always producing tryptophan when it's not needed, it is a waste of energy and resources for that bacteria. But that tryptophan, once produced and once in abundance, can bind to the repressor, and that repressor is considered to be activated. And then that activated repressor can go ahead and bind to that operator, blocking the RNA polymerase from transcription. So again, like I said, repressible means that repressor is inactive. Okay, RNA polymerase is constantly making that tryptophan. Once that tryptophan is abundant, that tryptophan molecule is then considered a co-repressor, binding and activating that repressor protein, and that repressor protein can go and bind to that operator, blocking RNA polymerase. So now let's take a look at the inducible operon. First, we're going to read that definition and then think about the English. So an inducible operon is one that's usually off and not expressing genes. So let's think about this word inducible. It describes the repressor, right? The repressor can be turned on. Okay, so again, this operon can be turned on so that the default is off. And so what does it mean for the operon to be always off? It means that the repressor is bound, okay, to the operator as a default. And so this repressor is said to be inactivated in order for that operon to be, you know, turned on. So what turns on this operon? Well, a molecule in the environment that's considered to be the inducer binds to the repressor, thus inactivating it. And then when the repressor is inactivated, the repressor is taken off of the operator, and RNA polymerase can then transcribe the genes. So let's recap. That inducible word describes that ability of the repressor. So again, the repressor is always on the operator, thus default for that operon is off. We are not transcribing anything. And so in order for it to be induced or to be turned on, the repressor must be inactivated. And so the repressor gets an inducer to bind to it, thus inactivating the repressor, and that allows for the RNA polymerase to transcribe the genes. The biggest example of the inducible operon is the lac operon. So what's the function of this lac operon? So lactose needs to be broken down, and it's broken down by three enzymes. And the generation of those three enzymes come in this operon. Okay, the presence of lactose increases the amount of allolactose, which forms as lactose enters the cell. And so this allolactose acts as an inducer in activating the repressor and thus turning on the operon in order to produce the enzymes to digest that lactose. So let's see how all that works. So inducible means to be able to turn on, right? So turn on. So default for this lac operon is off. So that makes sense. No lactose is in the cell. It doesn't need to be digested. So no enzymes are being made. But once lactose enters that bacteria, allolactose, an inducer, again, binds to the repressor. And so that repressor cannot bind to the operator, and thus the RNA polymerase is allowed to go and transcribe the three enzymes in order to break down lactose. Okay, so this is only when lactose is available. So again, those enzymes will be produced by this operon because it's turned on. Because the default when lactose is not there, the repressor is considered to be active and is allowed to attach to that operator, thus blocking transcription. So again, default is off, that repressor is on, right, or active, and then the inducer binds to the repressor protein, thus inactivating it, thus turning on this operon, allowing RNA polymerase to transcribe the three enzymes to digest lactose. So we can think about the operons in terms of metabolism. Inducible operons usually function in catabolic pathways, coding for enzymes that break down or digest large molecules. And again, these enzymes are only made when the digestible molecule is present, or the inducer, right? They can become the inducer binding to that repressor, turning the repressor off, which causes the operon to be turned on. So the other one, repressible operons, still consider negative control. Remember, we're talking about the repressors here. The repressible operons usually function in anabolic pathways, coding for enzymes that synthesize or create larger molecules needed by the cell. So the enzymes stop being made only when there's high concentrations of the larger molecule present. And these larger molecules act as the co-repressor, binding to that repressor and then activating it to turn off the operon. Okay, so again, the repressor will be activated by the co-repressor, thus binding to the operator, turning off the operon. So we have talked about the negative control operons, but there is something called the positive control of operons. And these are controlled by things called an activator protein. So when the regulatory protein is activated, it stimulates the transcription of an operon by increasing the affinity for RNA polymerase. So what does that all mean in simple English? So once activated, the activator can bind to the DNA and then that attracts RNA polymerase, thus increasing the rate of transcription. So the biggest example would be cyclic AMP receptor protein, the CRP, and sometimes they're called SCAP. They are considered to be the activator proteins for this positive control and specifically for the lac operon. So many genes and operons are controlled by more than one regulatory mechanism, so usually a combination of both negative and positive, and that applies to the lac operon. So we know that the lac operon is considered to be an inducible operon. But what is also cool about the lac operon, it has a positive control aspect. So when glucose, the preferred food source of E. coli, is scarce, the lac operon is stimulated by the binding of CRP. So again, let's recap there real quick. For this positive control, glucose is low. Again, the lac operon is stimulated because CRP is activated. The cyclic AMP levels increase when glucose resources are low, and this cyclic AMP activates the CRP. And so we see here visually that again binds to this inactive CRP. Once cyclic AMP binds to the inactive CRP, it activates it. This attaches to, the location right upstream of the promoter, and then this attachment to the DNA attracts RNA polymerase. Okay, so when glucose levels increase, CRP detaches from the lac operon, thus decreasing the transcription of the lac genes. Okay, but this only is again true when there's the presence of lactose. And in this case, allolactose will be generated and binds to the repressor, thus again turning on the operator. So let's recap that because there was a lot of information thrown at us. So the lac operon is considered to be the inducible operon. Okay, inducible means that it has the ability to turn on. So our default is off. And the only way for it to be off is if the repressor is bound to the operator. So how can we turn it on? So again, the presence of lactose. Okay. So when lactose is in the environment, this again creates the allolactose to bind to the repressor, and then that will activate the transcription of the enzymes to break down lactose. But there's a positive control. Okay, so the positive control is when glucose is low, CAMP increases. Okay, that binds to the CRP or cap, and that activated CRP binds to this CAP site and thus attracts RNA polymerase to this DNA operon. Okay, and then transcribe those enzymes. So the combination of both lactose being in the environment and glucose being low creates a faster turnaround of the breakdown of lactose. So again, a lot of those enzymes that break down lactose is created. Okay, but when glucose levels are high, again, this affinity decreases and then it returns to normal transcription of lactose if lactose is present. It's not a very high or fast rate of transcription. So again, it work it works both ways, but for maximum production at a fast rate, lactose must be present, and then the activated CRP must be bound to that cap site to attract RNA polymerase to again transcribe the gene. Those two conditions must be there in order for maximum production of these enzymes that break down lactose. And then finally, we have some genes that are always expressed, and then they're always on no matter what. And we see this image here. This is usually true for proteins or RNA that is always in use by the cell, aka ribosomal genes. So those would be considered always turned on, and there's no regulation to turn them off. And then we have reached the end of the slide. Hope this helps y'all. Okay, have a good one.